Welcome to Time to Talk Science and Medicine. This podcast is brought to you by the Translational Institute of Medicine at Queen's University, Kingston, Ontario. My name's Dr. Charlie Highmarch, and I'm an assistant professor and researcher at Queen's.
And my name is Stephen Archer, and I'm a cardiologist and researcher, and I'm also the director of the Translational Institute of Medicine here at Queen's, which is called TIME.
In this series, we're fortunate to talk to science and medicine with some of the most accomplished translational researchers from Queen's University and beyond. We want to know what their dangerous idea is, which is a concept they're working on, which if true, would change the direction of research. But we also want to find out a little bit about the person behind the research. In previous podcasts, we've sat down with clinicians, basic scientists from Queen's and beyond, to discuss a diverse range of research, including cancer, cardiovascular, bleeding disorders, and the impact of animal care on human and animal well-being.
Today's a very special podcast, and we've invited Dr. Will Sharp from the University of Chicago. Will's a friend of mine, and he's an emergency room doctor there. He's an associate professor, and he divides his time between looking after very sick people in the emergency room in South Chicago, around University of Chicago area, with the conduct of basic science. And he has approached cardiac arrest and how to resuscitate people more effectively as a scientist would, while at the same time actually performing cardiac resuscitation on people with the disease. So he both sees the problem and addresses it and tries to find the molecular underpinnings of cardiac arrest and use those to develop new therapies. So we're delighted to have Will visit. And before we get into the science that you're doing with all this interesting stuff about cardiac arrest and mitochondria, I'd like to hear about your origin story. How did you become an emergency room doctor and clinician scientist?
First, let me just say thank you for inviting me here. It's just a great pleasure to come here and see you again and to share my work with you. So as to your question, my origin story is I grew up in South Carolina, and my father was a geologist. So I sort of grew up on the campus of the University of South Carolina, and I always knew I wanted to be a scientist, but I didn't really know what type of scientist because I didn't like rocks. So I wasn't really interested in geology. So I sort of was exploring the science aspect of things, but then I became interested in medicine through being a Boy Scout. And I taught first aid and emergency preparedness with an emergency medical technician who really encouraged me to think about the health sciences, and then a visit to the gross anatomy lab in high school. And I knew that like healthcare was something I was interested in. And so in undergraduate, when I was finishing, I decided I wanted to go get my PhD in cell biology, but it was at a medical school where I would be interacting with medical students and future physicians. So that really sort of, I thought I might combine my love of science with sort of interacting with the medical profession. But as I trained these future physicians by teaching them histology and gross anatomy, and they were going into the clinics and really telling me some fascinating stories about their patients. And I really realized that I really missed interacting with people the way I did when I was a Boy Scout and camp. And so I really wanted to go back and do my medical degree. So that's what I did. I went back to medicine, got my MD degree at the University of South Carolina, and then, of course, was interested in emergency medicine, and so did my residency program at the University of Michigan because they really were encouraging people to become physician scientists. So that was a good, and then found my way to the University of Chicago, which at the time was very encouraging for physician scientists.
That's interesting. So I have to ask you, has your father ever gotten over your failure to embrace his love of rocks? That's a hard one for your dad.
Yeah, I think as a parent, you're always hoping that your children will be somewhat interested in what you do. And I can tell you that my two children have no interest in medicine. At least that's what they're saying right now. So it's always a little bit of a disappointment. But we're each our own individual.
Yeah, and it's interesting. Like, just to give me a time frame of this, because it's easy to say, "Oh, I did my PhD," but you got into medicine through doing some serious science, and then you later brought science back into your practice of medicine. But how many years did you spend getting that PhD and becoming a legitimate scientist? Says he with no PhD, I might add.
Yeah, I think that was some of the students asked me that same question earlier today. I did a sort of a traditional PhD program. So instead of some people do an MD PhD program, which is very sort of condensed. You walk into the lab and they say, here's your project. If you complete this in the next 2 or 3 years, you'll walk out of here with a PhD. Mine, I showed up in the lab and my advisors were like, well, this is what we do. It's up to you to come up with your project. And so that's a big leap for a lot of people. And so I spent 5 years doing my PhD. And then at that point, I kind of knew that I wanted to go to medical school, but I needed a little bit more time just to kind of get out. So I did a postdoctoral position after that.
That was, how many years was that?
Oh, so I did a postdoc for 3 years.
So that's 8 years so you were well prepared.
Yes. I was
well prepared. And then some.
So given that you have obviously an extensive research background and you have the emergency medicine training and doctoring, how important do you think that the interplay between those two kind of fields is to your current position?
Oh, I think it's extremely important because I think scientists and physicians think a little bit differently at the end of the day. Scientists, there's no black or white. Everything's gray. And where's the evidence? What is the evidence pointing me to? And you always are questioning the evidence. So there's just always uncertainty, and you realize that. I think as a physician, we naturally tend to be more black and white. And like, your patient, when you're talking to your patient, the patient's like, am I having a heart attack or am I not having a heart attack? It's not like, well, maybe you're having a heart attack. So they really want, am I having a disease or I don't have the disease? And at the end of the day, you've got to make a critical decision. And I think that way of thinking is a little bit different. So I think it's been helpful that practicing medicine, I understand that there's a lot of things that we don't know about medicine that we often take for granted. So I'm always questioning the evidence, but at the end of the day, I still have to make the best medical decisions that I have.
Yeah, and speaking of evidence, there's evidence that my brain is not working properly, because one of the things I forgot to mention when I was introducing Dr. Sharp as an academic, a researcher, is that he's also a leader of emergency room doctors. He's the president of the Illinois College of Emergency Physicians, so he has that as well. And he gave a stellar time medical grand rounds this morning. And maybe before we get into any kind of specific questions, because Charlie's question just reminded me, you're at the bedside, so you see the evidence. And one of the things that struck me about your talk this morning was the comment that something like only 5 to 10% of people that have an out-of-hospital cardiac arrest actually survive long-term. So I wondered if you could just give us an idea, what is the magnitude of the problem of cardiac arrest, either in America or the world, and what are the success rates, which I guess inspire your research?
Yeah, so it's a huge problem. The estimates vary, but some will say 500 to 600,000 arrests every year in and out of the hospital. My emergency department, we probably have 1 to 2 cardiac arrests coming in every single day. Some of the arrests are never even transported to the hospital because they have protocols for terminating the arrest if they think that the outcome's not going to be good. So a lot of patients arrest in the field, and we're not even seeing them as physicians. But the patients that the emergency medical services resuscitate, at least temporarily, and then bring to our emergency department, I'm probably seeing 1 to 2 patients every day in the emergency room. So the magnitude is really large. And a lot of times in the emergency department, we resuscitate these patients, and we feel really good about ourselves. We pat ourselves on the back thinking, oh, we just saved a patient. We did a great job. But the reality is these patients go up to the intensive care unit, and our ICU colleagues really know that a lot of these patients don't do well. So it's a sobering thought.
So is the 5 to 10% success rate accurate? And if so, are these 5 to 10% of people actually going back to work, or are they just surviving but in a diminished state?
Yeah, so I think it depends on which denominator you're using. The 5% is probably like everyone who's arresting. The patients, and we know that people who arrest in the field, about 40% of them survive to being admitted to the hospital. So my perspective is on the 40% that are surviving to the hospital. And of that 40% that get admitted, we're seeing probably about 30% of those people being discharged. So 30% of the 40% are being admitted, and then about half of those have good neurological outcomes. So, but again, when you're looking at the total magnitude of the disease, the percentage is very small.
Yeah.
So as Dr. Archer said, obviously you gave a great talk this morning where you sort of elaborated on some of the evidence-based ways that you're maybe changing that kind of care. Now, on this show, we actually like to ask our researchers what their dangerous idea, which really is us asking, How would your hypothesis kind of change or disrupt the field of study as it is if it was true?
Yeah, so I think that there are a couple of concepts. I think the first is that a major concept in the field is that people have their cardiac arrest, and that is when the injury occurred. And then once we get them to the hospital and we've resuscitated them, it's a matter of just simply trying to modify that injury. But the injury has already occurred. There's not much we can do about it. And I would say I think that one of the concepts I'm trying to get across is that may not be necessarily true for a lot of our patients, that the injury is actually progressing in the minutes to hours following the resuscitation, and that there's a critical window of time to intervene on this patient. So I think that's one of the first concepts I want to get across. And then the second concept is really this idea of, so the oxygen that we breathe every day in the room, it's not toxic, right? Our mitochondria detoxify it. We don't consider it hyperoxia, which is like abnormal. We know that like too much oxygen for anybody is dangerous. That if you breathe 100% oxygen for too long, you'll go into seizures. It can certainly cause some pulmonary injury. And certainly in neonates, it causes eye problems. So we know that oxygen's toxic, but because our mitochondria do such a great job of detoxifying it, we don't see those problems. But I think there's a growing knowledge that oxygen may be toxic in critical care illness. And what if your mitochondria are no longer functional? If your mitochondria are not functioning, the oxygen in this room now becomes toxic. So I think the idea that mitochondrial dysfunction and oxygen toxicity are sort of interesting topics in the critical care field.
I think that's a transformative way of actually viewing mitochondria and the role that they do in sort of like metabolizing this toxin effectively to use it to make it something useful. So obviously in your talk this morning, you were proposing actually, rather than giving too much oxygen, maybe reducing the amount of oxygen you give to a patient in order to reduce the injury in that critical window.
Right. We to avoid confusion, we call it oxygen restriction therapy. Although we're literally, in our animal model of cardiac arrest, we take these animals and we resuscitate them, and we put them in an environment where they're only receiving 10% oxygen, which is probably sort of being like on top of Mount Kilimanjaro. So it's physiologically possible. It might be stressful, but it's certainly physiological possible. And when we randomize animals to receiving different amounts of oxygen, whether it's this oxygen restrictive treatment, which is 10% oxygen, or normoxia, which is 21% oxygen, or even mild hyperoxia, 30%, the outcomes are vastly different in a critical 6-hour window following resuscitation. So it really is time dependent, and it's oxygen concentration dependent.
And without getting all mito geeky on people, I mean, this sort of relates to the fact that I think from listening to your talk that oxygen is used two ways by mitochondria. There's the normal way where oxygen is like the catcher's mitt at the very bottom of the electron transport chain, and electrons are transported to molecular oxygen. And that's a healthy response. It doesn't generate toxins. And then in cardiac arrest, you showed very nicely in your talk that the electron transport chain is sort of blown to bits and is dysfunctional. And then the oxygen, as I understand it, is changed in a way that turns an oxygen molecule into an oxygen radical. And those radicals are called free radicals that go off and damage the tissue, if I understand you correctly.
Right. And I guess the simplified way of thinking about it is it's like a nuclear power plant that's no longer working, and you're continuing to feed fuel rods into the plant. And now you're just getting free radicals produced, sort of like a radioactive poisoning or oxygen toxicity.
And so I guess by inference, if you called, instead of a code blue, a code mitochondria, people could come running with mitochondrial therapies to shut off the radical production and give you a longer window to resuscitate the patient.
Yes, that's the idea.
Yeah, says every geeky mitochondrial biologist ever and no physician ever. Okay, well, so you've done a good job explaining your dangerous idea to Dr. Highmarsh, who also has a PhD, but now let's turn it around. If you were trying to explain this idea about how to preserve the window for resuscitation or how to resuscitate people better to a high school student, how would you put it in sort of lay terms?
Yeah, I think the idea is that again, oxygen is a gas that we take for granted. But can have certain toxic elements if given too much or in the wrong context can be toxic. And so, particularly in critically ill patients where their mitochondria are not working, oxygen now becomes toxic and is causing more pathology. So I don't know. That's what I think the message I'm trying to get across.
And it's interesting, I guess context really matters because oxygen is good when you're healthy at the normal level. But oxygen in the specific state of the few hours after a cardiac arrest is different because your mitochondria are damaged. And in those hours, what would have been an acceptable amount of oxygen suddenly becomes a toxic amount of oxygen for the damage you've sustained kind of idea, I guess.
Right. I think that's conceptually what we're trying to get across. And so matching the amount of oxygen to the patient's metabolism and their mitochondrial function, I think is critically important.
Perfect.
Okay, so maybe we can sort of move away from the high school and back to sort of science, and you can tell us about maybe your latest discoveries and how it will impact the clinic as you see it.
So I think like the first one we've been talking about is just that oxygen restriction appears to affect the outcomes in our model of cardiac arrest. But the second part is that there are maybe pharmacological strategies to intervene on this mitochondrial injury process. One of the molecules was this Dripitor 1A, which is actually developed here at Queens by the Dr. Archer lab. And so we've been able to use that drug to modify this process called mitochondrial fission, where the mitochondria are, I guess they undergo this fission division process after cardiac arrest and are probably contributing to the damage injury. And we find that giving this Dripitor 1A not only improves outcomes following cardiac arrest, but it may also ameliorate some of the effects of oxygen toxicity.
And I remember because you and I had the great pleasure of sharing lab meetings for many years at the University of Chicago, but you published some beautiful work where you showed in cardiac cells the mitochondria breaking into pieces, which was a nice illustration of fission. And you used different strategies before there was a Dripitor 1A drug. You had other strategies I think you used to counteract this phenotype, if I remember right.
Right. We had MDV1, which is another inhibitor of mitochondrial fission. We also tried cooling or hypothermia. And then we also, I think we tried some calcineurin inhibitors as well.
And more recently, you've even used drip knockout mice where this mediator of fission dynamine-related protein 1 is missing. Those mice, as I recall, are protected?
Correct. Yeah, I didn't really show that in the talk today, but we have a DRIP1 knockout mice that, and when you do ischemia reperfusion on them, their hearts are protected. So we know that this DRIP1 mitochondrial fission protein is a critical mediator of injury following cardiac arrest.
At this point, I think it's probably just worth asking the question, in these models that you're translating to mice, can you just tell us about the resuscitation of a mouse?
Which doesn't get enough attention.
Which doesn't get enough attention, but it's one of those curiosities. No. And as somebody who's worked with animals for many years, I have been responsible for doing compressions on small rodents. But maybe you can tell us a little bit about the model, because that's important for us to understand how it might translate back into the clinic.
Sure. So, I mean, obviously these mice have to be surgically prepared, so they get anesthetics. So we know that's one sort of difference between our animals and our patients. These animals sort of get anesthesia to begin with. And then once we put an arterial line catheter in them and we hook them up to the ventilator, and we also put a central venous catheter into their internal jugular vein to administer a potassium chloride dose to induce the cardiac arrest. And then what we do is we actually sort of turn off the anesthetic so the animal is starting to wake up. So we try to limit the effects of the anesthesia, but we basically turn the anesthetic off. And as the animal's waking up, that's when we push the potassium chloride, and it puts the animal right into cardiac arrest. And we then know that the animal doesn't have a blood pressure, it's not breathing, we disconnect it from the ventilator. And then the EKG tracing goes into an asystole rhythm, so there's no electrical activity in the heart. And then we wait for different periods of time to resuscitate the animal. So we might wait 4 minutes, we might wait 8 minutes, or 12 minutes. And typically 12 minutes is the optimal sort of length of time that we look where we get the most injury, but animals are still surviving. But at 12 minutes, we will start cardiopulmonary resuscitation, where we start doing CPR in animals. And we do that for about a minute and a half to kind of replicate what a patient would receive. And then we give a dose of epinephrine that is comparable to what a patient would receive in their resuscitation.
And when these mice kind of recover adequately, they are identical to control mice that have just received a sham, or are there neurological deficits? Or
these animals are very ill following resuscitation. In the first hour, they're often hypotensive. Their heart function is not as good as a normal mouse. And then we wait for about an hour for the animal to sort of start recovering, breathing on its own, so we can we extubate the animal. We take it off the ventilator and allow it to sort of recover. A lot of the animals die in the first few hours, and then the ones that are recovering, they have neurological injury. They're not moving. They're not grooming themselves. They're not moving around the cage. They're not exploring. They really show neurological signs of damage, and they have delayed responses to neurological testing.
And these does this kind of replicate the situation you see in the clinic in humans? Is it a good model for people?
Yeah, it's probably in some ways milder than what we see in a lot of our patients in the intensive care unit, because patients are typically on the ventilator. They're not going to be extubated right away. Patients are going to be on the ventilator for days before you think about taking them off the ventilator. With these animals, they have to come off the ventilator pretty quickly, because we know that mice develop ventilator-associated injury, and two, just from a practical standpoint, we don't have a mouse ICU in which to monitor mice for days on end. So we cannot exactly replicate what our patients are going through. But so I would say in some ways our model might be even milder than what our patients are seeing.
And just to give our listeners sort of a sense of the magnitude of, if you were right, how big the effect would be. So if you don't do anything and they have a cardiac arrest and you do CPR and get them back, what % of those animals die with no drug intervention?
If we do no drug intervention, we probably have a 50%, like just normal cardiac arrest resuscitation. We're probably seeing like 50% of the animals die pretty soon. Whereas with the drug interventions, a lot of times it's up into the 80 to 90% survival. So it's a lot dramatic. Yeah.
And so those drugs that you've tested, because having listened to your rounds, which I realize that our listeners haven't, but so you talked about several drugs where you intervene specifically to stabilize mitochondria during that resuscitation period by giving a single shot of a drug. So maybe you could just tell us briefly about a couple of the kinds of drugs that might, if they could be used in humans, might be able to increase the survival rates with cardiac arrest.
Okay, so one was targeted at this electron leak concept that we were talking about earlier. So it's this S1QEL that came out of Mike Murphy's lab. This S1QEL can be administered right after CPR, and that seems to be very protective for the animals. The other strategy was the Dripator 1A, which was developed in your lab. We also find that giving that immediately following the cardiac arrest is protective, but it may also have protective effects if we wait like the first hour after arrest. And again, it also seems to be protective. So there may be this critical window of injury which these pharmacological agents will act and improve outcomes.
And I guess that leads to the thorny question for every clinician scientist. It's easy to fix a mouse or a rat, but just how do you even do research in people that have had a cardiac arrest? I mean, you can't talk to them, you can't get consent to do a research study. So if you wanted to test oxygen restriction in patients, or even more, if you wanted to test these drugs, how does one even begin to approach that when the patient is dead or almost dead?
Yeah, for me, I think one of the next strategies too is just investigating some of these effects in like larger animal models, like a pig model or even a rat model, to make sure that they translate well. But for your question for patients, it really is, we are fortunate in emergency medicine now to have this trial network. It's called the Siren network. It's a network that's really linked all these different emergency departments together to do these processes, and they are developing processes of exemption from informed consent where they could just start these interventions, particularly if they're very promising. And they have an educational process where they inform the community about these very promising events. So I think the future is promising because we are developing the infrastructure for emergency medicine trials.
Yeah, I guess if you think about it, no one asks you whether you want CPR. I mean,
no, it's true.
I mean,
I do consent to it. I just want to say that online.
It's sort of, you might pre-declare that you don't want to be resuscitated in a hospital, but if you're just out in the community, it's sort of considered the compassionate thing to do is to try to resuscitate someone if they're found down on the ground.
Right. And one of the students in your group today asked me, like, well, how do you get consent for cooling or for hypothermia of patients? And I said, well, that's now sort of considered standard of care. Now, the actual level of cooling is sort of up to debate, like whether you cool the patient down to like 32 or 33 degrees, or whether you simply modify them so they're not getting a fever, 36 or 37 degrees. There's a wide range, but cooling is considered standard of care, and you don't have to get consent for that.
Interesting. Well, I hope the drugs move on to clinical trials so they can actually help people, hopefully, and same with the oxygen restriction. So you've obviously been pretty successful. You're an associate professor at one of the leading universities in North America, and you've got several NIH grants, but every scientist has bumps in the road where things don't look so great. And I'm just curious if you'd be willing to share with us any bumps in the road, perhaps to inspire others who may be having a bump themselves.
Yeah, I think we all know that their funding is a big issue for science, and getting the funding is a hard thing. You're passionate about your science, but you've got to really communicate your science well and get others passionate, particularly study sections when they're reviewing it. And they want to see what's the big idea. How is this going to change the field? And I think you've really got to practice that. And so I, after as an emergency medicine physician, I was pretty busy with COVID. Certainly have personal and family issues through COVID, as well as just working in the ER under those conditions. And so I think funding for me became very low, and actually my lab was running out of funding. So that was my bump in the road: is how do you keep your science going? Because it takes a long time to get these grants. I mean, to get an NIH grant, you write it, you apply for it. I mean, that's like a year-long process. And if they turn you down, they say no funding, well, what's your backup plan? So, that was a really struggle for me as I was running out of funding. And I think that the way I was able to weather that is, first, I had great collaborators. I had not only from first the psychological support, having, people telling you the science you're doing is great, don't give up, keep going. I think that is, so you got to have that cadre of colleagues who just march you along. And the other part is I actually had people who came to my financial assistance. They said, hey, I think we're doing some similar work. Let's collaborate. I'll fund your lab technician for the next year. Even though they're doing work for me for stuff, we'll kind of help them do some work for your studies to get your preliminary work. So I think having good and the environment you're in, good colleagues is just essential for science.
I think one of the common themes that we come out of the bump of the road question is research funding. And one of the common answers that kind of stitches all of the podcasts we've done together is the power and importance of mentorship and collaboration. And you certainly substantiate that, the idea that kind of, we are in this together. We're all on the same team trying to answer the same questions.
Yeah, and it is interesting, I think, again, for listeners, because when someone has 2 NIH grants and is publishing well, you think it looks easy and you think the person's successful. But as I know, as your friend, I mean, the latest grant you got, I believe, was actually triage, or as we call it in Canada, streamlined in one round, and then you revised it, and it came back and scored at what percentile?
8th percentile.
Yeah, so it went from being not good enough to be fully discussed to being the 8th best grant out of 100. And I've seen that time and time again. The problem is when you get that triage or streamlining notice, you feel, I can say this myself because I've had this happen to me. I mean, you feel like, am I barking up the wrong tree? Am I misguided? Am I a bad scientist? And so it is nice to have people around you to validate that you're not an idiot. And it is worth persisting.
So thanks very much for kind of discussing that. The bump in the road is always kind of one of those kind of hard questions to ask, and it's a hard answer to hear. But Let's move on to kind of good times and where we're going to go from here. What's next for your work? What's the next step here?
Yeah, I think we really need to understand this critical window of injury following cardiac arrest. Is it like how long? Right now, our therapy, we started about an hour after the arrest, and we go to 6 hours. But could we extend it? I think that's how long is this window that because sometimes patients may not get resuscitated or get the therapies they need for several hours after they got resuscitated. So is there how late can we wait before we start giving these therapies? And then I think the other is can we combine therapies? Is there an ideal cocktail that we put together, or do we limit oxygen along with these cocktails, and do they work in larger animals? So there's so many unanswered questions.
I mean, I and even as you're talking, I'm kind of thinking of ideas, and it's your research program, but actually, here's a question. Do you see sex difference?
Yeah, I know we're doing that right now. We've not seen differences, and we put male and female mice to these protocols, and there has been this reported effect that women might have better outcomes from cardiac arrest, but at least in the mice that we've been studying, we've not seen that difference.
Right at the end of your rounds today, you mentioned something interesting relating to heterogeneity or diversity, which is that there's different causes for cardiac arrest, but the resuscitation is currently conducted by physicians is largely generic. It's done the same for everyone. And you were saying although as a cardiologist, I tend to think of people having cardiac arrest because they've had a massive heart attack, but you were saying that's not the predominant phenotype or cause of what you're seeing at University of Chicago. Who's having cardiac arrest, and does that matter?
Yeah, I think the etiology is vitally important, right? Because the pathology is different in how you treat them. We know that, it's obvious, patients who have hemorrhagic shock from their being shot or stabbed, those patients are going to have a very different outcome and treatment approach because, cardiopulmonary resuscitation is not necessarily as effective if you don't have any circulating blood volume to circulate when you're doing cardiopulmonary resuscitation. But our big problem, we see a lot of fentanyl overdose. We have an epidemic right now of opioid addiction and opioid overdose. And so fentanyl is probably a major contributor, or polysubstance abuse is contributing to our cardiac arrests. And those arrests are probably more like respiratory in nature than sudden, arrhythmia of the heart. So these patients are probably hypoxic for a time before they go into arrest. And so is the pathology different? And how we approach the treatment of these patients going to be different. And so that we're trying to address that right now. We have we just developed a fentanyl overdose cardiac arrest model for which we got some funding for. So we're right now comparing the outcomes in the fentanyl overdose model to our potassium chloride model.
With the idea that fentanyl may be having some direct cardiac effects in addition to suppressing respiration.
Fentanyl having direct mitochondrial effects even. So.
Yeah, I know Dr. Shetwan Zhang here at Queen's University is also studying the effects of fentanyl on ion channels in the heart. So there is, although it's remarkable, people stop breathing, that's what you see. But there's, in addition to that, with big doses, there's also cardiac effects. So very interesting. Before we wrap up, though, we've talked a lot about mitochondria, the queen of all organelles.
Have we talked enough about mitochondria?
I don't think one can never say enough about mitochondria. But you've satisfied my need for that, at least for today. But we have a few questions so we get to know you a little bit more about the person behind the research. So we're going to ask you some questions for which there is no wrong answer. So the first question I'll start off with is, what is your favorite meal, and can you cook the dish that you like yourself?
My favorite meal. Yes, probably my favorite meal is I love, my wife gives me a hard time. She thinks I should have been Asian. I love rice. I eat rice all the time. And obviously, all you need is a rice cooker. So I know that's a kind of a boring answer, but I eat lots of chicken and rice, just sort of dishes that I mix together when I come home after work. And so I'm a big rice eater.
Good.
Okay, so a new question for the podcast. You get to test drive it. If you could time travel, would you go backwards or forwards, and why?
Oh, that's a fantastic question. Gosh, I have to choose. I would love.
You do.
I would love. I mean, part of me would love to go back and do rounds at the bedside with William Osler. I think he's just a really inspirational, and a Canadian, by the way. Probably one of the greatest physician humanists of all time, and to be at the bedside as he evaluates a patient and his thought process. Boy, I would give anything for that.
Just go meet your heroes.
Yeah, meet some of the heroes. But the future is very intriguing as well.
At least you'd be back before the election. So we're not allowed. But you don't have to go.
Was there election?
There was an election. I heard
it was in the news somewhere.
I'm not sure it worked out that well. So what's the strangest job you've ever done? Other than emergency room doctor, we'll consider that a normal job. But
normal job, I had a summer where I had the summer before I had done a work at a marine biology station, which was a lot of fun. But probably the most down-to-earth job I did is the summer after that. I wasn't sure. And I got a job working for a propane gas company. And I went to people's homes and checked their propane gas tanks. And was it intellectually stimulating? No, not necessarily. But I met people from all different walks of life, meeting people and their trailers and then their mansions. I mean, all these people are using natural gas and, interacting with them. I learned a lot about just people and just how they live and seeing them because you went into people's homes. So, and as physicians, we don't really do house calls anymore. So we don't see the social environment, but it gave me a window of insight into, I think, how my patients are probably living in their conditions that I don't necessarily see. I saw that working for the propane gas company. And so I think that informs me. So I would consider that a very informative
And you're still involved with the idea that gas is important. So there's always consistency that you can find. Okay, so this is the possibly the most important question you're going to get this week. And we're going to close with this. And if you were a cartoon character, which one would you be and why?
That's so easy. Are you familiar with it? And so I guess in the 1960s or 70s, there was this cartoon called Johnny Quest. And I would come home, my friend and I, we'd come home after working in the lab all night, 5 a.m. or something. We'd come home and we'd turn on the TV and the Johnny Quest was on. And to me, I was like, oh my gosh. So Johnny is this young kid and he's like the hero of the show. But for me, the real star was his father. His father is Benton Quest. And so Ben Quest is the scientist. He has his own private island. He has his own private laboratory on the island funded by the federal government. He doesn't have to write grants. They just give him money. And he also has a supersonic jet. And if they need to go anywhere in the world, they just get on their jet and they fly. And he solves the world's problems through science. And so he's constantly getting, and of course there's always nefarious characters around the world causing different troubles. And he is using his science to solve the world's problems. So if I had to choose, I would probably be Benton Quest.
I think, didn't Johnny have some fabulous gun that he carried? I think there was a Johnny Quest gun that was popular with kids. It was sort of it looks like an AK-47. But anyway, well, that's a good answer. Well, maybe we hope you get your private island, maybe with a hollowed out volcano.
Unlimited federal funding within my lab. And it was in the South Pacific, so it was really nice.
Oh, perfect.
That's nice.
Perfect. Dr. Sharp, on behalf of Dr. Archer and myself, I'd like to thank you for taking time to talk to our audience today about your research and your discovery and your thoughts of science in the future. Also to our listeners, I'd like to thank you for taking the time to get to know Dr. Sharp with us. We hope you enjoyed the show. If you've enjoyed this episode and would like to know more about research at Queen's University or to share this podcast, you can search for Time to Talk Science and Medicine wherever you normally look for podcasts. Thank you very much.
Thank you. It's been a pleasure.